The next presentation, presented by Ben Aviner and Grossman, be presented by Ben Aviner, and it's the title is Augmentation of Calcium Influx in Cultures of Red Cortical Neurons at Hyperbaric Pressure, and now we are dealing with much lower pressures.
So I probably do not need to explain to this audience about the high pressure neurological syndrome, but in short, professional deep sea divers who descend to depth of 100 meters and more may experience many signs and symptoms, including vision and auditory deficiencies, impaired coordination, and cognitive and motor disturbances. Other signs and symptoms include dizziness, nausea, changes in EEG, sleep disorders, tremor, convulsions that can develop to full-on seizures, and even loss of consciousness. Now, this constellation of signs and symptoms is being attributed to some kind of neurological malfunction. Now, that malfunction can be manifested whether within the nerve cell or in the communication between two nerve cells. This communication is the synaptic transmission, essentially a chemical process between two nerve cells that is designed to deliver an electrical pulse from the presynaptic neuron to the postsynaptic neuron. This chemical process begins with the entry of calcium into the presynaptic terminal. And it has been shown in the past that synaptic transmission is generally depressed under pressure. Thus, it is of interest to examine the calcium flux into the presynaptic terminal at pressure. Already more than 20 years ago, Grossman and Kendig have done a research in the lobster showing a large depression of the response to stimulation under pressure, as you can see here, in the excitatory junctional potential. And more interestingly, that depression could be mimicked by reducing the extracellular calcium concentration by half. And this is one of the first works to demonstrate the relationship between calcium concentration and pressure. And it has been a long time desire of Professor Grossman to perform calcium imaging under pressure. And unfortunately, we had the opportunity to do so in the past year. We did these experiments of calcium imaging in a rat primary cortical neuron culture, which means we would get dishes about the size of a 5-shekel coin or a quarter, with the cover slip covered with neurons. And these neurons were incubated for 20 to 30 days in artificial CSF. And it is important to note that in these neurons, the literature states that about 50% of the voltage-dependent calcium channels are of the L type, and about 10 to 20% are all the rest, the T, N, R, and PQ, and that detail will become relevant later. We then loaded these cells with a dye, fluor-2, which is color, and what's nice about it is that if we project a certain wavelength light on them, just like a camera flashlight, they would emit light back on us, and specific wavelengths are 340 and 380 nanometers. Of course, nothing will happen unless we would stimulate the cells. We can see them, but we also need to stimulate them. We did that with an electrical stimulation, and to our knowledge, this is the first time in these cultures to stimulate electrically. And this is all very nice, but nothing would, that means nothing unless we measure the emitted light, and we did that by ratiometric measurements, meaning we would take 2 snapshots approximately per second with the 2 wavelengths. And what's nice about the fluor-2 dye is that as calcium concentration rises, the light emitted from the 340 nanometer wavelengths is increasing, and the light emitted from the 380 is decreasing. And it is the relationship or the ratio between these 2 wavelengths that actually tells us what is the concentration of the calcium in the cell. So after I bored you with the details, we can move on to see some nice pictures. This is a by 10 magnification movie. And as you can see, these red hot flashes are elevation of the calcium concentration following an electrical stimulus. This is the same frames just repeated in a loop. After we verified that it actually works, we could zoom in to a by 40 magnification. This is a somewhat slower film. You can see these hot red colors, meaning an elevation of the calcium concentration. We could then designate specific region of interest ROIs, within the frame using a special software. Approximately 20 somata and 20 dendrites were picked in each experiment, sometimes more. I'm saying dendrites, although the actual term should be processes, because we cannot really tell whether these are dendrites or axons. But for simplicity, I just call them dendrites. This is a typical screenshot. On the left, the panel you just saw, and on the right, the measuring panel. For each one of these regions, we can measure the baseline or the basal calcium concentration, actually the basal ratiometric emission, and then the peak following the electrical stimulus and a slow decay. Again, just typical. If you follow one of these RIs, let's say the dotted circle, you can see a pre-stimulus cold colors, and then after the stimulus, elevation and slowly decaying back to control levels. These all experiments were done in Jay Dean's lab in Tampa, Florida, in the Nautilus chamber, quite a big one. In the middle here, you can see the microscope mounted inside the chamber. It might be difficult to see, but over here there is a radiator, which is very important in order to keep the temperature constant while we compress the chamber. And over here in the middle on the table, that's the actual dish having two stimulating electrodes. Of course, a perfusion and a suction of the artificial CSF, and also a heater, which allows us to keep the temperature constant while decompressing the chamber, in order to verify that our results are not due to temperature changes within the compression and decompression. And in the middle, you can partially see the porthole of the microscope. So let's see what we have found. Compressing to 0.3 megapascals, the equivalent of 20 meters of seawater, what we witnessed was a significant increase of the ratiometric emission in both somata and dendrites. Thus, we can say that the calcium concentration peak augmentation was augmented by high pressure. This correlates with the results from experiments done in our lab not too long ago by Tal Palau, which we'll be talking later on today, in which a reduction of the field excitatory postsynaptic potentials, meaning an input for a given neuron, were reduced at pressure as expected, but the output or the population spike for that neuron has remained unchanged or only slightly reduced, meaning that the relationship between the output and the input of a given neuron has increased. Thus the term increased transfer function. And the question was, what was the source for this phenomena? And a possible explanation could be a dendritic or somatic boosting of the currents, and I've just showed you that it actually was measured in our experiments, calcium imaging experiments. So we wanted to verify whether this augmentation is stable, and we have found that it is not stable. You can see here that somewhere between 5 to 10 minutes, the augmentation has decayed and became non-significant, meaning this high-pressure augmentation was transient. This also correlates from experiments done by Grossman and Kendig almost 30 years ago, in which increased action potential currents were found. As you can see here, an augmentation due to 50 atmospheres compression, but in the course of 20 minutes later, that augmentation has been reduced even to lower than control values. The same happened in the action potential velocity, a first increase and then decaying to, to levels below control. Then we started to notice something else. If you concentrate on the constellation, constellation over here, you can see the colors became more and more and more red. But this is not following a stimulus, but the first frame for each experiment, meaning the basal levels of calcium concentration, the baseline. In 1 atmosphere, 3 atmospheres, after 10 minutes, and after 20 minutes, meaning that there was an elevation in the basal calcium concentration. As you can see in this figure, compression to 0.3 megapascals has significantly increased the levels, and further compression to 0.6 megapascals, the equivalent of 50 meters of seawater, has further increased the levels. Each point over here represents about 20 ROIs in both somata and dendrites. Decompression did not recover the levels back to control. Thus, we can say that high pressure induced an increase in the basal calcium concentration. This increase was not transient or reversible, and this increase may be caused by the cell membrane depolarization that would allow more calcium to flow into the cell. And this was also suggested, or actually measured in the same work by Grossman and Kendig. As you can see here, a depolarization of the cell membrane as pressure increases. This work was done in a lobster. I already mentioned the 0.6 megapascals, so let's see what we have found is that a reduction, slight reduction only in the dendrites right after compression, but this effect has disappeared already 5 minutes later, and the levels remained at about control for the next 30 minutes. So this is a bit confusing. There is no calcium concentration peak augmentation by high pressure? I believe a possible explanation for this phenomena could be that once the initial augmentation has subsided after the first compression to 0.3 megapascals, we do need to remember that there is more than one type of voltage-dependent calcium channels within these neurons. And after the initial augmentation has subsided, other channels that are depressed by the pressure has become more prominent, and the total effect we can see is levels at about control. Decompressing back to 0.3 and 0.1 megapascals in both somata and dendrite, it seems like it is a bit depressed, but it is not significant. Thus, we can say that it is either that the high pressure induced calcium concentration peak augmentation is reversible, or, as I just stated, that the total high pressure effect on the various voltage-dependent calcium channel types is manifested as a recovery to the basal calcium concentration. So, to summarize that up, high pressure, hyperbaric pressure has induced an augmentation of the calcium concentration peak. This augmentation was transient. Higher compression did not further augment the calcium concentration peak. And we witnessed an elevated basal calcium concentration by pressure. Future work would need to concentrate on screening the specific high pressure effect on the various calcium channels types by the use of blockers, and I can already tell you that the work has already begun. I would like to thank Professor Dean and Professor D'Agostino sitting here in the audience for hosting Professor Grossman and myself and allowing us to use their very unique equipment. I would like to thank Carol Landon, the lab technician, for helping us preparing these experiments. I would also like to thank Rafa, which took good care of us and also provided us with some social activities, as you can see here, or diving in freshwater rivers in Florida and also in the oceans. And of course also thank Professor Grossman for inviting me to participate in this research. And although he's already an emeritus professor, has performed many of the experiments by himself after I left. And also thank you for listening.